Hello everyone, today we'll be discussing about neutrophils and chemotaxis. Now, what is meant by chemotaxis and why is it essential? I have drawn a basic diagram showing endothelial cells, lumen of the blood vessel which contains neutrophils and it is part of the innate immunity. These are some smiling, healthy looking cells, and this is tissue resident antigen presenting cell. Here I have given the example of dendritic cells. Now, what will happen if there is an invasion by bacterium? It's gonna damage the cells, right? But how will the neutrophils come to rescue to the site of injury? So, there must be some sort of signals released from the site of injury which causes or attracts the movement of neutrophils towards the damaged site. This is literally meant by chemotaxis. Chemo mediated by chemical mediators and taxis is movement. This is divided into four broad steps. The first step is rolling. Second step is activation. Third step is adhesion. Fourth step is diabetes. Now, I would like to first start with the recognition of bacterium by the dendritic cells. Have you ever wondered how does our body know that this is a harmful bacterium or virus and it is something which needs to be killed, and something which is host cell and it is safe? So there are something called as PAMPs, which are pathogen associated molecular patterns. These are basically conserved sequences in the microbes which are repeating biochemical structures like lipopolysaccharides, flagellin, peptidoglycan, etc. So these things are absent in the host, only present in microbes, and our body has evolved to recognize these. So the dendritic cells will have certain receptors called PRRs or pattern recognition receptors. These pattern recognition receptors recognize PAMPs. When this interaction happens, the dendritic cell releases a lot of chemical mediators like TNF alpha, histamine, thrombin, and chemokines. Now, I'll be coming step by step how these mediators will help in the movement of neutrophils to the site of injury. First step, rolling. So, as we have discussed, these are endothelial cells. Now, under normal conditions, there are something called as P selectins, and these are absent on the cell surface, but under conditions of inflammation, when molecules like TNF alpha are released, these P selectins move to the surface of the endothelial cell. Now, why is this important? These P selectins will have interaction with neutrophils via PGSL1. I don't think you need to remember the full form, but it is p-selectin glycoprotein ligand 1. Now, when this interaction happens, the neutrophils, it's like putting a brake on the neutrophils. They slow down and they start rolling. Now, after rolling, the second step is activation. Now, what is actually being activated? As we have discussed, there are chemokines also which are being released. Example, CXCL8, which interacts with CXCR1 receptor on neutrophils. Now, when this interaction happens, it leads to downstream effects and various intracellular molecules and pathways are activated, which cause including talents and kindlins, which causes a conformational change in LFA1. This constitutes the activation. Now, why is this LFA1 important? It is important for addition. Now, sorry, I'm going to use red. Now, this is the blood vessel. These are endothelial cells. There are certain molecules known as ICAM1, which interacts with LFA1 on neutrophils. Now, this interaction causes adhesion and halting of neutrophils. Now, what is the next step? The neutrophils have stopped, but they actually have to cross the barrier to reach the site of injury. Right. The neutrophils have halted, then it leads to release of autotaxins. Now, these autotaxins, here is an interaction between ICAM1 and LFA1. Now, after this halting and the release of autotaxins, this autotaxins loosens the connections between the endothelial cells. Then the neutrophils can kind of squeeze in and reach the site of injury. So this is the broad mechanism of neutrophils and chemotaxis. There is a bacterial invasion. There are release of certain signals which leads to interaction between p-selectin PGSL1, which kind of slows down, then LFA1 halts, and then there is diapedesis, and then the neutrophils can do its job. That is it. Thank you.